Good day, class. For today's pre-lab discussion, we are going to talk about the first part of exercise number 10, the development of the chicken embryo. So, before we begin our discussion and the use of some pictures to point out anatomical structures and developmental landmarks, let's first examine or review a few things as introductory concepts. Now, let's remind ourselves that similar with amphibian embryos, chicken embryos are easily obtained. So you should not be shocked or you should not be amazed why so many books, why so many monographs are available, and these monographs showing the development of the chicken embryos. Now, because they're easily procurable, we have a lot of information, we have a lot of literature about them. Secondly, it's important to take note that majority of the developmental characteristics and changes of the chicken embryo are almost identical with other vertebrates, particularly of mammals. Hence, giving us a lot of advantage, a lot of benefit by studying chicken embryos first, because similar structures can be appreciated from the chicken embryo when we compare it or when we study mammalian embryos. No? So you will be able to appreciate similar landmarks that are positionally and functionally related with each other. More so, let's also remember that the chicken embryo undergoes a very different type of cleavage when compared to microlecytal eggs, that of mammals, and mesolecytal eggs, that of amphibians. Um, since the chicken embryo contain a lot of yolk, we categorize it as a telolecytal type or telolecytal egg. The cleavage pattern that it shows is very different. Now, so yolk has a retarding effect on cell division. That's why, in a way, if you remember in activity 4 in exercise number 4-6, you remember that when we open the fertilized egg, we see that only a small space is actually occupied by our growing chicken embryo. So the cleavage pattern is said to be meroblastic, and the embryoblast is displaced towards one side of the yolk, appearing as a small or appearing as a disc, a surface disc. So we remember that, or we can associate, we can say that chicken embryo cleavage pattern is discoidal and incomplete. Now, more so, it's also important to take note of the establishment of the animal axis before we look into structures that can be appreciated in the 24-hour, 33, 48, 72, and 96-hour whole mounts. Now, so two important axes can be distinguished in the early embryonic stages of the chicken embryo or early developmental stages of the chicken embryo. First one is the dorsoventral axis. So we know that since the animal follows a superficial discoidal type of cleavage pattern, one side of the growing embryo touches the yolk, and those cells are said to form the endodermal part, said to form the ventral parts of the animal, while the other side, the exterior, which is not in contact, not in any way in contact with the yolk, with the yolky cells of the animal, will become the dorsal part. Also, let's remember the development of another important axis at the same time, the craniocaudal or the anterior or antero-posterior axis of the animal. In lecture, we can review or we can remember that the antero-posterior axis of the animal lies perpendicular on the long axis of the chalaceae. If you